Welcome back, everybody. And per usual, I'd like to go ahead and start by just seeing where people are, how people are doing. Many of you may be done. We're in the last week here, and so we're actually still going, but we are hanging in there, and yeah, it's about as best as I can say. Go in order of when people popped in. So Rebecca, let me go to you next.
Yeah, we actually are finished ourselves. I submitted final grades earlier today. Yeah, it was a relief for sure. We went till Thanksgiving and then had remote finals. So that was how we did it. But yeah, glad to be done with the semester.
Zach, how about you?
We went online after Thanksgiving, and nominally our last day of classes is Monday the 14th, although I'm ending on Friday just because no one's going to learn anything on Monday. And then we have a week or so finals, and final grades are due, I think, on December 24th, which is super dumb. It's a terrible.
What day they do?
December 24th. It's really good idea. It's awesome.
Amazing.
But you know, so we have this week of classes, week of finals, then a week of grading.
So the registrar can input them on Christmas Day, right?
Probably, yeah. Maybe on New Year's too, if you're lucky.
Yeah. Let's go to Melanie next.
Hi, everyone. This is our last week of classes. Finals are next week, but I intend to wrap everything up by Monday. So I've been remote all semester. So finals and everything are online.
So nothing new for you there, which is good. Yeah. Alonzo.
Yeah, last week for us too. Finals grades go in on Monday. I didn't do finals this semester. I just did projects, so that made my life a little bit easier that way, and students too. But yeah.
Cool. And then finally, Ronia.
Hi guys. So I joined in a little bit late because I was putting in grades. So we've been remote all semester, but our last day was Monday, this last Monday, and it's finals week right now. So we're putting in grades and doing finals. And Alonzo, I might have to get more information about your project idea because I want to do that next semester. I'm not liking finals right now, so. Yeah,
just to quickly leap back to that, what kind of classes are we talking about there that you did that were project driven?
Yeah, so I got a research methods course, and so that made for a nice, easy project where they design and experiment with different techniques. And then the intro neuro course, we did exams through the semester, and then for the final, it was an integrated question. So I lied, it wasn't a project, it was an integrated question, but they came up with a scenario where they included concepts from throughout the semester and sort of included in one nice write-up. We did a Android spy that they had to create.
So not a 3-hour come in and sit down exam, which I think is the very good in terms of what you have to grade. Yeah. So with all of that said, and this actually gives us a little bit more, well, now the ample free time many of you have, right? The paper here. And this has actually been really cool for me because we have, again, 3 sessions for these journal clubs. And because of various delays, things like that, I haven't actually, I've talked about this paper with another group, but it's been a considerable amount of time since I've had to think about this. And there's a couple different issues in play here. It's not a very long paper. The reason I had picked this one out was less about the specific content and more about the opportunity to talk about a couple broad ideas that this paper invites. One was using the COVID-19 pandemic as a teaching tool that a lot of people have done to various degrees. I think a lot of people have used it for really cool engagement. In other cases, I think there's been COVID fatigue in some circles about everything in the world being COVID-centric. So I guess it depends how you apply that. And then the number 2 thing in here is the consideration about developing skills and sort of how does one take a current idea and use resources that are available online and that are developing, help your students to discern what is scholarly versus not scholarly, all that sort of stuff, and design projects around that that are actually going to support the type of biology ideas that you're looking to advance. So there's sort of the general idea about how to contextualize some of this and then how to effectively do it. In this case, it was immunology majors at UAB. So how would you do this for different audiences? So I think that's where a logical point for us to think about this takes place. Sort of how have you, not necessarily COVID per se, but how have you guys used current events or events, items that are maybe quote unquote real world useful for your students? And have you been able to find ways to use them in your own curriculum? I can speak to some of the stuff that we've done. It's been mostly taking COVID papers and using them for journal club, discussion of ideas, things like that. Again, I sort of struggle with how to best present primary literature, which is sort of its own beast. But getting students to get excited about current work by showing them papers, literature that is brand new, and exposing from that the idea of what has been done effectively versus what might get improved. And so I'm gonna go to Alonzo next for his comment there about students hating it.
Yeah, they just didn't like it. I mean, I try to start off the semester. We have guest speakers come in, and we have so there's real labs that they can join. And a few students are successful after a speaker comes in and joining those labs to do research in the lab based on what they've learned and things like that. But overwhelmingly, the students don't like it. I try to give them like an article worksheet to sort of help them digest it. And we talk about how all science papers are very complicated to some degree, but you have to have these techniques to sort of filter through and find what you're looking for. And overwhelmingly, at the end of the semester, they all still hate primary literature. Like they appreciate the structure that I provide for them learning to read it. And I think that this is one of the ways to maybe make it a little bit palatable. But we can't always be talking about things that are so relevant. And so once you get to more complicated techniques, often I've found that the students seem to push against it. Although that said, I think one of the things I need to take into consideration is the comfort that we have as senior people in science with dealing with not knowing everything. The students get really frustrated by, and so I don't know when they did this intervention. It was my first thought was, did they throw the students in here in week 2 or 3, or was this after they'd covered everything already? Which was one of the main things the students were asking for was, there's so many techniques in here that I don't understand. Can we talk about these before you have us read the paper? Which isn't the way science happens, right? We're all reading stuff we don't understand, but for undergrads, I think that that's something that at least I've taken away they're looking for.
Yeah, there's a larger, and I should have spoken to it more right here at the beginning, context of how do you guys go about incorporating primary literature into your courses. This is something I certainly struggle with in terms of not even getting them excited about it, because that's a bridge too far, but getting them to understand how not to be intimidated by it, like how to be comfortable with questioning what you see in a scientific paper and offering critique in a scientific paper. I don't do this at all with my intro students. Some people do. Our intro students are a mix of majors and non-majors, and I don't really want to get into the weeds with this with students that like aren't necessarily even going to continue in the field. But for that next level up where I do get people who are going to be science majors, I try to maybe take a paper and look at it over not consecutive sessions because I get sick of it, but like, you know, once a week, let's talk about these particular figures. Let's talk about how you build an argument, how you build a criticism, so sort of ease them into the process. But I'm always intrigued with people's ideas about how they get around this issue of this being daunting for your students. And so what other ideas have people utilized for this?
I had kind of a similar thing to add on to Alonzo, and I found some very similar results as well. Last semester, so spring 2020, we got kicked online in March, and I happened to be teaching a comparative vertebrate zoology class, and COVID, of course, is a zoonotic disease, as are a lot of these potentially pandemic causing diseases. And so I thought it was a pretty interesting opportunity to talk about why are we seeing this rise of zoonotic diseases, what characterizes them, is it going to get worse potentially as we continue to encroach into habitat that previously people weren't inhabiting? And I found for that class that they really liked it. Like they would have like a weekly discussion, but I cheated for that particular class and I said, okay, here's some journals to pull from, and you can pull like editorial comments, or you can do reviews, or you can do like little pop sci intros, briefings, as opposed to having to do full research papers. So they loved that because I think they were really curious about COVID, and especially then when it was hot and it was fresh. It was an easy way to force themselves to look up new information about it and sort of feel like on the front lines. For other classes, so for example, I teach an upper division herpetology class. Herpetology is the study of reptiles and amphibians, just in case anyone doesn't know. And we read primary literature once a week for that class. And it's very funny because most of my students are environmental science. And when it's something that they feel comfortable with, even if it's a 20-page paper, if it's on maternal nesting behavior, some aspect of ecology, they really like it. They're like, oh, this paper was so inspiring. It was so fun. It like put off all these ideas. And I do give them structure. They have like a, worksheet they need to fill out as a part of it. But the minute we start moving into something maybe they're a little less comfortable with, so like I'm a physiologist, and so of course, as we follow like the more physiology heavy units of that class, they get, I think, uncomfortable, but the way it presents itself is they are bored, and then they'll write in the final reviews for the class like, I really liked the papers, but I wish you had chosen more interesting ones. Like I liked half of them, basically. Okay. And it's like, well, the point of it was to link the papers with the topics as we were covering them. So like we do the urinary system and then read a urinary paper of some kind relevant in the field. And probably some of that is me. This year was my second year teaching that class. So it's like always working on trying to improve things, tighten it up, but maybe they didn't quite see the link I was trying to get there. Like, oh, first we intro the content so that you're supposed to have more tools available to you to read this paper. I suspect even when I'm teaching this class for the 100th time, they'll still say, yeah, this was too dense.
Well, yeah, so you made them do work, that's a given, so yeah.
So yeah, I had to, and I don't know how to fix that. Like, I'm not gonna throw out all of our phys papers just because they don't want to do it.
Yeah, nor should you. I am curious, as you're describing this, what is the frequency of new papers? So are they doing a paper a week, or what is the...
Yeah, that's right. And that's actually something I've considered too, is like, well, I actually am asking them to read a lot. Maybe I should cut it down a little bit every other week, something.
Yeah, I mean, that was sort of my first, and I'm not even arguing necessarily there's something to fix, but if you're thinking about trying something different, there could be a less is more thing here where you do fewer papers, but you spend more time talking about them, like maybe, a couple different sessions on a single paper. One session talking about what the paper says, another one like building on it or something like that.
Yeah, that's a good idea.
Yeah. That's intriguing. And then what level class is this?
So it's a 300 level. And that is, the expectation is that they're juniors, seniors, and they've taken all of their GE.
But these aren't intro level students. They know what they're doing. Okay, yeah.
So speaking of intro level students, that's where my issue is. So I've been contemplating, and as I said to Alonzo, I hate like having to do exams right now because of the level of dishonesty too. These students, I mean, I found my exam on Chegg for my organic class, and I get so agitated when I'm grading because I know some of the answers. Like, I'm like, you don't know this. This was something clearly Googled. So
you're talking about online examinations?
Yeah, because we have to deliver. So I've been trying to come up with an alternative method. And I know this is somewhat related, but I like this idea of real life application, not necessarily to the primary literature. And I've played around with the idea before even COVID is giving them research papers for introductory level courses as well. But I teach chemistry. So my dilemma is this, and I don't know if you guys can help me. I have a certain level of content that I need to present.
Yeah.
So I need to do this, and that, so that when they go to the next level, my colleagues are like, what has she been teaching them? You know what I mean? Yeah. So that's my number one dilemma. The other is, and I want to be able to test them. I think we can learn a lot from traditional testing, but my dilemma is like, I want to give them case studies. And I found this, it's called let me show. I've used it before, and maybe National Center for Case Study Teaching and Science.
Oh, sure, yeah. They're based out of Buffalo, correct?
I think so. And I don't know if you have to pay for it. I used it a couple of years ago, and it was free on the teacher side.
I don't think you do.
Yeah. So it's really cool. They have like 75 case studies just in Gen Chem, and they have for organic. And instead of the primary literature, because I want to give them real life, real world applications, but I just don't know how to do that without information overload, without really, I think I already kind of overworked them with the homework, the online homework and the content, and giving them additional material like primary literature or case studies. Do you guys see that as making it more difficult for our students at such a difficult time?
So what do people think about that, about sort of figuring out the proper balance? The content versus experience thing is fascinating, and figuring out how to best do that. You mentioned organic chemistry.
Yes.
Is that the course that's involved here?
So yes, I teach the fundamentals, general and organic. So first year, second year courses. Mm-hmm.
So what do people think about that? I have to say, I haven't heard a lot of people talking about, and it's useful to have you here as someone of a different discipline. I'm less familiar with people talking about this in chemistry than in biology, of the importance of presenting primary literature. And I'm wondering if I just don't know, are there certain papers you can use for those things that highlight some of the techniques you're looking to show? That might be too simplistic, right? Because you're trying to do these foundational things, and these papers by definition are probably going to go further than that. I guess for me, and I'm just thinking about it from my own biased perspective, I would be more inclined to do that kind of stuff, not waiting till junior senior necessarily, but like with intro chemistry. I don't know necessarily if I would try to do that. That's just me. And then when you get sort of these intermediate students, can you provide a couple landmark papers that maybe you could do, again, a less is more approach, like just try a couple of these and do some like in-depth discussion, not only about what the content is, but also how you're supposed to read it. There is the create method, which I have not actually used myself, but I know it is well received elsewhere. And Alonzo, you look like you're going to say something.
Yeah, so actually my POM mentor, she used it in her class that I was sitting in on, which was for seniors. It was like their seminar class before they graduate. And yeah, it was really in depth. I really enjoyed the process. I think it's like almost 2 weeks per, or maybe even 3 per paper. And it was a series of papers, but it's about a concept. And I thought it went really well. I think to your point about, trying to do that or something similar to intro course, it might be a bit too far, but the create process, I actually thought it was pretty excellent. And
yeah,
I don't know if it would work for every course, but it definitely has a lot of merit to it.
And I'm putting, I just put the URL for it if you want to read about it in more detail. And a lot of it is based, again, with biology classes in mind, but I really don't see why it wouldn't work in a science class. Sort of like chemistry, you've given the world POGIL, and there's no particular reason why POGIL can't work in other plans, but it's definitive. Like when I think of POGIL, I think of it as a chemistry thing, right? That's done definitively in a chemistry environment. But the same concepts are there, right? That I want to do an active approach or an engaging approach to get the students to think about classes. I think all this is really interesting because, again, the paper we have in front of us was done with immunology majors and sophomore immunology majors. So I'm at a small school. We wouldn't have an immunology program to begin with. But you have people who have already sort of bought into a research science or medical science themed model here. And so you're not working towards that. You're not working to get them excited about biology in general. Now you're trying to train them up for skills, right? As a skill set. And actually, that's another point as I'm talking out loud. The purpose of why you want them to read scientific literature in the first place. And for me, it's certainly not like, oh, let's learn about these particular phenomenon. For me, it's about how do you learn to read and interpret these papers. I've actually been very gratified to hear positive feedback in places I wasn't expecting. People preparing for MCATs, things like that, because it turns out with the way the MCAT is now drawn up, the ability to read and interpret these passages is directly related to that like sort of same skill set. So I guess if you think about it in terms of like, what is your point? Like, what are you trying to do? That might guide sort of what levels you would do this at, how frequently you would do it, etc. I appreciate like the content you have to get through though, right? Like it's you have less flexibility, I think, than a lot of us in terms of some of our choices.
I would just offer one thing that I did this semester. It was, I have a challenge too. I teach at a community college. Most of my students coming in are not familiar with scientific literature, primary literature. I taught a 200 level cell molecular course completely online. And I was really scrambling to try to put together something coherent. And one thing that I found, at least showed them a figure, if not the entire paper, right? I used like that TRIM method from one of those life science education papers, where I just would take like a figure from a paper. And what I really enjoy, I love podcasts. So I tried to find something relevant. So Michael, you mentioned something that's relevant real world. So tried to find a podcast that was relatively recent or relevant, and link the podcast to a scientific paper.
Cool.
And then I called it a model. I would use these for their weekly discussions. So they would have to like read the model, which was just, several pages, and included sometimes a figure or two from a scientific paper. So they would listen to the podcast, link to the model, link out to relative websites that supported that concept. And then it was a discussion. So it was an online discussion because this class was asynchronous. So then I posted some like questions for understanding within the model, and then they would post their answers to those questions on the discussion board, and then they'd have to interact with other students. So it wasn't really reading the literature or understanding it critically necessarily, but it was at least just introducing them to the fact that we got to this podcast because of this paper or something recent in the news. And this was some of the evidence that supported that.
That's neat on a couple fronts. I mean, I've certainly had colleagues that have tried to employ podcasting in their classes. I have never ventured forth into that, but it seems really cool from the outputs I've seen in some other contexts.
Yeah, I mean, I'm sorry, Michael, they didn't have to create them. I gave I gave them the link to a podcast to listen to that podcast, but
Yeah,
I do like,
right? The idea of bringing in another media to use. I think in some classes I'd like them to make them and really try that out. But actually exposing them to these are primary papers, but like getting a podcast and weaning information out of a podcast would certainly be useful to your students as well. Especially, what level students were they again?
So this is a 200 level cell and molecular class at a community college.
So that's a good opportunity. Not ground level, but not, super advanced either, to get them thinking about these concepts in a different way. I like that.
One thing I'd like to add on to that, I really like that idea, and that's something I've not seen before in a course, using podcasts, having them listen to it and sort of going through this like structured worksheet. I bet they got a lot out of it. I have a colleague who does kind of a similar thing, but sans the podcast. So he'll, it's a molecular genetics class. So it's, upper division, 300 level students. And what he does, which might not be appropriate for 200 level students, I'm not really sure. I think it's kind of a gray zone, but what he does in that class is he'll start them off with like these sort of tailored worksheets, more like what I'm thinking you're describing. And he'll do that for part of the semester. And then after week 6 or 7, we have like a 13, 14 week semester, depending on if it's a COVID year or not. He has them present papers. So they pick and then present and are supposed to, the idea is like they've learned the foundation of how do I pick tools from a paper, and now they should be able to, in groups, present papers on these topics. So that might be an interesting way to add to, maybe take them up for the last week or two in like full synthesis of like, okay, how do I really read a paper after I've gotten this like lovely foundation for how to read a paper. I've never done it myself, but I've always thought that was a really cool thing that he does. So
that's a great idea. That would be my projects, right? With the answer to the finals. That would be a great project.
So Michael, I teach I teach Gen Chem pretty frequently is my consistent rotation, and I've tried to bring in some science cases into Gen Chem before from the Buffalo site, and it did not go well. It was not the right time. It was hard to apply, and it wasn't just the loss of content. I've gotten over that one a long time ago, right? You always lose content. It's just the way of things. It was that the students had a hard time figuring out what they were supposed to do. They didn't really have a lot of context for it. It was not obvious why they're reading papers or why they're looking at case studies or what they're trying to get out of it. Maybe it could have been structured better, but I found it to be not as helpful as I thought it would. But I also teach an upper division for juniors and seniors. It's called Biochem too, but really it's a journal club, and we meet 3 times a week, and all we do is read papers. That's the entire point of the course is to read primary literature. And over the time that I've taught it, I've cultivated a series of 15 to 20 papers that are usually older papers that are applicable and useful. Well, not really applicable, actually, they're not applicable at all, but they're relatable. The students can figure out the terms in them, and they can understand the experimental logic. And we talk about why we're reading it and how to pick apart a paper. But invariably, every semester, I try to bring in 1 or 2 modern papers, and it doesn't go well at all. They're too complicated. It's too many things upon things upon things. And sometimes I pick a good one, in which case I add it to my rotation. But most of the time, the newer papers they're really hard. They're really dense. They involve techniques and ideas they haven't seen before, and we'd have to spend weeks digging through it. So I was impressed with this business. I haven't looked at the paper this paper references, whatever it's called. Where is it? Is it in the methods? No, there it is. Sorry, on 2.3, distinct immune response in 2 MERS-CoV infected patients. Welcome me. I haven't read this paper, and so I'm curious as to what it presents, because I've found, again, that cutting edge modern stuff, at least in the biochem field, is extremely hard for students to get a hold of. Older stuff is easier, it's simpler, but it also doesn't mean anything because it's published. I have a great Search paper from 1980. It's a lovely paper, but the students weren't even vaguely alive in 1980. So it's this recurring theme, which is like approachability versus relevance, and I struggle with it a great deal.
It's funny you say this because I teach a class that I nominally call medical genetics, but really is sort of what you're describing. Let's learn how to read primary literature. And I let the students pick some of the papers. And one of the papers that we utilized this semester was written in 1986. And they presented it to me, and I'm like, oh, okay. Like we actually don't use half of these techniques anymore. And the stuff they were doing over the course of an entire paper would have been like 1 figure in a modern paper. But wouldn't you know it, it actually was pretty cool in terms of going through and learning how to dissect like primary literature in the way you describe. So I think I sometimes am distracted by the idea of getting the shiny thing, the brand new thing out there, when sometimes these classic papers do work. Sometimes when I'm looking for a candidate paper to use, I will go into PubMed or something like that, and I'll type in sort of the technique I want to highlight and see if I can get something to pop up. It's not the most efficient way in the world of doing it, but sometimes I find some gems hidden out there. CRISPR, good example. Well, what's a good CRISPR paper? And so some of them I've found have been great. Others they've gotten lost in all the other stuff they're trying to do. So that's an interesting approach, like taking some more classic things. I also like what you said there in terms of like, if you find something works, you sort of can file it away for future use. It was effective at the time. So you don't worry then either. I mean, that's Biochem 2. It's not a content heavy class then, it's an experience heavy class.
It is a 0 content course. Okay. As a matter of fact, it is designed for that. And so we have nowhere to go and nowhere to get to, which is really remarkable. And the person that designed it doesn't allow us to get anywhere, right? It is very firm that it is for the reading and understanding of primary literature, and no like structured core content can be taught. And so the literature often has nothing to do with it. What we're doing on Monday is totally different than what we're doing on Friday. The topics are different, the techniques are different, but you have to be reading biochem literature and getting after it. And I wouldn't mind if it were a bit different than it was, but just because reasons, that's not an option. So it is what it is, and I try to do the best I can within the structure. We read a lot of papers, and we talk about a lot of papers, and there doesn't seem to be terribly much burnout. We're still at the end of the semester going reasonably strong like we were in the beginning of the semester. The burnout comes when I pick a bad paper. Usually one of the modern ones I try to throw in there. It's just a wreck, and because no one gets it. And then they get, like, I think Alonzo was, I don't remember who was saying this, they get angry, and maybe it was you, Michael, they get angry and frustrated because they don't know things, and then they it just spirals out of control.
I was curious about the content because and this goes with what Roni had said as well. In my own experience, biochemistry is one of those, like, you got to get these certain things done for all the people that want to take an MCAT exam. And I have always struggled when I end up teaching biochemistry, how to balance my pre-med population with my, shall I just say, general population in terms of like what I'd like to do versus what I know I kind of have to do in content-wise. So that sounds really cool, the way you do that. It sounds like you're very explicit about what the class is, which sounds like a really good thing for your students. Alonzo, go ahead.
Yeah, I had a question for Zachary about the biochem course with the papers. And it's one thing I was wondering about this paper as well. So with the methods course, similarly, we had a pre and a post test, and I saw some improvement there, which you expect. They just went their whole course about the techniques. They were pretty favorable about the assignments and the engagement that were similar to this, the group learning about these things. But in terms of the assessment, right, where we're talking about that anger and that frustration with the complicated papers, I tried doing exams, and even we had not just the paper, but we'd have the paper's senior author come in to give us a talk about it. And they were still so just not like, what am I supposed to know from this? We were trying to get them used to hearing these expert level talks. So I wanted to ask Zachary what sort of assessment tools you guys had for that class.
Yeah, so depending on how it's taught, sometimes I do a question set after every paper, and sometimes I do a question set after a couple papers, and we read a couple and then digest them. And they're all short to medium answer free response questions that I write, or whoever's teaching the course writes. And we try to not make it what's going on in figure 2, because we don't think that's a particularly good question. We try to make, how could this technique be applied in this case? What if they'd found this instead of that? How would you interpret the business? And we try to make it, if not open paper, at least we'll pull the relevant figures from the papers. They don't have to start memorizing stuff. And again, I'd be happy to send you some of the papers I do and some of the questions we write. I mean, I'd be very content to do that if you're curious. And the answers stay pretty solid, pretty consistent. These are junior, senior level biochemistry majors that have been through Biochem 1 already. So all that content stuff has been covered in the other class. I teach Biochem 1. So these students they're all there for a reason. They know the business, and they're all pretty intrinsically they're self-motivated to get after it. And we try to keep, I think last semester, I have to remember how I did it. I think last semester I was doing 2 or 3 papers and then a problem set, and then that sort of maintained throughout. And I found the results to be pretty good. The students were able to tackle primary literature as long as it was picked extremely deliberately. And that's been my primary issue, which is finding the right stuff. Some things I think are great aren't work, and some papers I don't like very much turn out to work really well. And I haven't gotten that sweet spot yet. I'll see if I can post something in chat. I don't know how to do that, but maybe there's a file attachment. Yeah, I'll play around with it.
I actually had a question for you too, Zachary, just based on what you just said. Deliberate picking of the papers, and it could be that and I assume there's some amount of this that is true, we don't quite overlap in what we need our students to take out of a paper. But I'm curious what you are looking for when you're picking a paper. Like, what about the paper do you say, oh, this makes it a great fit for this class?
Yeah. Well, when I find it, I'll get my Sea Urchin Binding paper, not mine, it's published in 60, but I'll get the Sea Urchin Binding paper. It's a lovely protein biochemistry paper. It's this really beautiful series of experimental progressions. Mostly what I'm getting is a paper that they can look at the figures and at their level, understand why is the figure done, what question was the author asking, and then how does this figure move their model from point A to point B to point C to point D. It's a paper that the data and results are interpretable and sort of comprehensible. And if the paper goes beyond that, it just doesn't go very well. So I focus almost exclusively, and again, I'll see if I can fiddle. I'm frantically looking through my business now. I'll see if I can find these puppies, because whenever I stumble across them, whatever way that is, oh geez, oh boy. Okay, I got to put myself on mute. But when I find a good one, it works really, really well. And I'll see if I can dig one up here for you.
I know when I have to think about, for instance, showing students what a bioinformatics or like a genomics data set looks like, I go looking technique first, and try to find in the figures things that will most carefully illustrate or demonstrate what I am trying to get out there. So I actually found, in turn, like to Zach's point of finding a paper I like and I keep using it, a paper that was at the very beginning of RNA sequencing or total RNA sequencing, that's not medical. It's actually about hibernation patterns in squirrels of all things. But actually going in, it's very specific about here is how I collected all the RNA and here is how I refined it. And then there's a black box in the middle for like some of the actual processing stuff, right? That'd be a whole separate issue. But then here's how I analyze the data, and then other people, you could talk with your students. Well, clearly other people could go in and use this data to do other things. And so I arrived there by simply trying to find something that would effectively talk about the technique, and then finding, seeing if the figures would support me in that. Like it would be good, useful for the students. Yeah, it's not the most exciting paper in the world, but it serves me two purposes. Number one, it's very useful technique-wise, and number two, every now and then I try to remind the students that not everything in the world is clinical. And so, I've got some ecologically themed papers or physiologically themed papers, right, that I can put out there as well. I see Zach figured it out there while we were talking, so. Okay, other.
Oh, no, I was just going to mention, I don't know if anyone uses Science in the Classroom, the annotated papers from AAAS. I have used those for my general biology course. Those are first-year students in the second semester. And I use, for example, the coral bleaching one for my general biology 2 course, which is ecology and evolution. And that annotated paper has embedded in it resources from BioInteractive from HHMI, and also, some resources like to support, evaluating that paper. So I found that the annotated papers, that one in particular, because I haven't used many of them, worked well for an introductory course. And again, we don't read the entire paper, but we do look at figures and, analyze some of the data within that paper.
That's a really good suggestion too, the annotated papers. I've not used them to a great extent, but the resource for that, especially if you're trying to get deconstruct like the scariness behind some of the process, those annotated papers are really effective for that. So thank you for sharing. All right.
And I just had to mention, I'm sorry, it's just funny that you're talking about classic papers, because like I used to teach, we used to have a biotech program at our college, and I used to teach something like what you're discussing, where you're just doing literature, evaluating scientific literature. It's not a content course. And I went back to those classic papers because when I did my PhD, 1,000 years ago, we were looking at one gene at a time and stripping the membrane and using another probe. And there were these, I was in, my research is in cancer biology, and there were these set of papers on the HER2 receptor amplification, the genomic amplification. And those classic papers, those early papers where they show a southern blot, they show, the intensity of the band, it's just so easy. It makes the interpretation of those figures so much easier than, like you said today, where we have these huge data sets and genomic analyses that are much more difficult to interpret. So I definitely can relate to what you're saying.
Yeah. All right. Like I said, there's not a lot. I mean, this is a good conversation starter paper. There's not a lot of data to this, but it's more about like getting us sort of thinking about this question. So I appreciate all the suggestions you all have shared with each other here. Does anyone have any sort of final thoughts?
I just wanted to say congratulations to everybody. You made it.
Almost.
So,
made it yeah. We're running out of time to completely screw it up. So, and Rebecca, you look like you're about to say something.
Yeah, I just wanted to, I thought this was a really useful conversation in terms of like ideas for helping me pick papers and potentially restructure my classes. And the idea for using older papers too, now that it has been said, actually makes a lot of sense to me. But I think I am victim to the like, oh, what's the new shiny thing?
And that there's something to that too, by the way. Like for me as a molecular biologist, geneticist, whatever, CRISPR-Cas9 is really important now, and I need to not only figure out how to teach it, but how to present it to my students. So shiny new things can be important sometimes, but sometimes these old papers like actually do a better, as Zach refers to, do a better job of explaining some of these concepts than the newer ones. All right, well, thank you guys for coming on a busy time. I will send out soon a generic doodle poll so we can figure out when people are free in the spring to do these sessions. But otherwise, have a good holiday, and we will see you next year.